Good afternoon, everyone. Good afternoon, dear candidate. Welcome at Maastricht University for a very special occasion, an exciting occasion. Hereby, I open this academic ceremony in which Tom van Mulken will defend the academic thesis, Robotic Microsurgery in Plastic and Reconstructive Surgery, the journey of creating, introducing, and preclinical validation of a new and dedicated robot for microsurgery. Quite a long title, Mr. Candidate. Before we start giving the floor to you, Mr. Candidate, I would first like to introduce the supervisory team sitting on the other side. Professor René van der Hulst and Professor Rutger Scholz and Dr. Q Xiao. Welcome. You have worked very hard in guiding the candidates towards this very special moment. And I will introduce the candidates of the opposition committee when they will propose their question. Mr. Candidate, please, the floor is yours.
Thank you. Highly esteemed Pro-Rector, dear colleagues of the Corona, dear friends and family and colleagues in the audience. It's a true honor to present our work of creating, introducing and validating of a dedicated robot for microsurgery. And my presentation will contain 4 subjects. First, I'll try to explain why we need a robot for microsurgery. Then I'll demonstrate the new robot. I'll elaborate on the studies that we've performed and the future perspectives. So first, the why. Why do we need a robot for microsurgery? And what is that to do with plastic surgery? Well, plastic surgery evolved from history as a reconstructive field. So even 2,000 years ago in India, already total nose reconstructions were being performed by using the tissue of the forehead. And techniques improved, but the real specialty of plastic surgery emerged after World War I and II, with the reconstruction of burns and head injuries. So what do we do in plastic and reconstructive surgery? We restore form and function. So if you're born with a defect like a cleft lip or a congenital malformation of the hand, a deformed hand, we can correct it. Or if during life you injure your hand or your face in traumas, we can repair it. Or if because of cancer treatment, parts of your body need to be removed, like breast cancer or skin cancer, we can restore form and function. And how do we do this? We relocate tissue, and that means we use healthy tissue and we move it to the problem area to solve the problem. And sometimes the tissue that we need is far away from the place where it's needed. And then we need to take the blood vessels attached to this tissue, we transplant the tissue, and we reconnect these blood vessels so the tissue that we transplant gets blood again and survives. So for example, I can take a muscle from the leg and put it in the arm to correct someone who's paralyzed, but we need to reconnect these blood vessels so that tissue survives. But we also need to reconnect the nerve, so this muscle actually works. And this is all very small. So these vessels are small, the nerves are small, and the sutures we use to connect it are small. So we use microscopes to see what we do, and they can augment our eyes. And it works very well. And that's why it's called microsurgery. We need microscopes to see what we do. And we can see very well, but our hand has to do the job on its own. We have some tools in our hands, but for the rest there is no help. And even though we are trained as microsurgeons, if we had a bad night of sleep, or a stressful situation, or a long operation, this shakiness in the hand gets worse. And every human being has a little bit of shaky hand. So no matter how hard we train, there is a limit in precision of our hands. And that's the why. That's why we need a robot for microsurgery, because these microscopes are getting better every year. Still have to do the job. And we're now at a point that we're operating on such a small vessels that we can still see them, but our hand is not coping anymore. And the robot can help. So that's the why. So our story started in 2007 when the Da Vinci robot came into our hospital, and it's a big robot weighing about 500 kilograms, and it's designed for operations in the abdomen or in the thorax. And we tried to do a microanastomosis with this robot. Professor van der Hulst tried it, and we concluded this machine is too big for microsurgery. It's too powerful. So together with the Technical University of Eindhoven, we decided to start a project to create a robot for microsurgery because there was no other robot around for a decade. So the concept of this new robot was that we wanted superhuman precision of the hand. But for the rest, we didn't want to change the whole operation room. So we wanted a compact robot that's compatible with the existing tools that we have in the operation room. So the existing microscopes, they can be used. The existing instruments that we have in our hands are very fine and they are reusable. So we wanted to click them in the robot. And this is the robot that we designed. So it's a small robot, and you can see there's a ring that can be placed above the patient, and on that ring there are two robotic arms. I can show you here. And these arms, they can be loaded with any microsurgical instrument that we want, with the use of 3D printed adapters. The surgeon is holding the joysticks here. They look like forceps that we used to have in our hands, and the robot copies our movement. And we get precision in two ways. First, tremor filtration. So the shaky hand, that signal is filtered out by the software of the robot. Second way is motion scaling. So if I move 10 centimeter, I can ask the robot to do the exact same movement, but just do 1 centimeter. And this gives precision and accuracy. So you can see a demonstration setup. On the left is our surgeon, Raimundo Cao. He's the technical engineer who designed the robot. He's sitting with an assistant as we are sitting in normal microsurgery. He's holding the joysticks in his hands, looking through a normal microscope. So you can see the setup doesn't change the operation room at all. So now to the studies that we've performed. So the first generation of the robot we only used in laboratory setting. And we did 2 studies, one silicone vessel study and one animal study. So the silicone vessel study is just silicone vessels. It's fake, but they resemble the blood vessels that we use to operate. And we asked 3 participants to do an operation to connect these fake blood vessels with small sutures. It was a microsurgeon, it was a plastic surgeon in training, and we asked somebody who never operated before, a medical student. And we compared the performance of the hand versus the robot, and we looked at time and quality that was needed. And what we found was that the time that's needed is much longer. So you can see here the robot time is much longer compared to the hand, but the time decreases very rapidly. Same for quality. So the initial trials, the quality of the robot was lower, but you can see you learn really quick. And then we proceeded to an animal study, because we had to prove that it's possible to do this in real living tissue, real blood vessels, real blood flowing. So we had to do a small trial of 8 rats, and we connected the abdominal aorta, which is about 2 millimeters in diameter, and even the femoral vessels, which is below 1 millimeter vessel. And that went successfully. So with these good results, we wanted to proceed. But then there was no budget to continue this research. So the Technical University of Eindhoven, together with the Maastricht University, they raised a spin-off company. And this spin-off company called microsurgery. And in this way, we could get new funding to continue the work. But it also gave conflict of interest, which meant the technical engineer became chief technical officer of microsurgery, and I became chief medical officer of microsurgery. And together with a part-time CEO, we started only 4 people to continue the work. But this gave me several hats. I was doctor, I was researcher, and now I was also director of a company. But this gave us the opportunity to continue the work. So a new robot was built with the lessons learned from previous studies, and we could continue towards human studies. And we did 3 types of human studies. So the first human study was lymphedema, Why lymphedema? Because that's the smallest and most difficult surgery that we can do at the moment. And lymphedema means that the fluids from your arm cannot go back to your body, and you get a swollen arm. And it happens after breast cancer treatments. So in breast cancer treatments, the breast gets operated, but also the axilla gets operated. And sometimes even radiotherapy needs to be added. And this gives scar formation in the axilla. And this scar formation, this blocks the fluids coming from the arm. And then you get a swollen arm, heavy, infections, painful. And it's a lifelong problem for a lot of women. So one of the solutions to this problem is to create a bypass of the lymphatic vessels, which contain all the fluids. And you make a bypass just before the area of the scarring to the veins, so the fluids can get out of the arm. But this, as I said, is very, very small. This is 0.3 to 0.8 millimeter in diameter vessels. They're tiny and they're fragile. So how do we do this? We inject a contrast agent in the hand, and we can track this with a near-infrared camera, and you can follow the fluids going up the arm. And then you can recognize the lymphatic vessels, and there is the area of the problem. This is where the scar tissue is, and we mark the skin there, and we create the bypass just there. So this is an example of an operation. You can see Dr. Kishau here on the left, also on the left. On the video, I'm in the middle there, Esther in front, and the robot's behind my back, because you don't need the robot for the whole operation. Sorry for this film, but this is real. Okay, so we look for the vessels with the microscope. They're tiny, and there it is, a lymphatic vessel. Then we take the robot, you can just rotate it in. You can see the ring and the robotic arms. Microscope comes down, and we continue. And here, Dr. Kishau performs the lymphovenous anastomosis, 0.3 millimeters in diameter. For the surgeons, 11 sutures, which is 5 times thinner than a human hair. So you can barely see these sutures with your naked eye. So this is an operation. And our study contained 20 patients, half operated by robot, half operated in the conventional way. And we looked at safety, efficiency, and patient outcomes. So what did we find? Again, we saw that the time it takes for the robot is longer in the beginning, but you can see a promising learning curve. It drops quickly. And the patient outcomes, more important, of these early trials is equal to the patient outcomes of Dr. Kishau doing it for already 10 years. So this was promising. Second study is robotic extremity reconstruction, which means your lower limb. So your lower leg doesn't have a lot of tissue around your shin. And if you break your bone in traumas, often the skin goes open, infections can come in, and the bone doesn't heal. And to solve this problem, sometimes we need to move tissue to place this on the fracture so it can heal. And we need to reconnect these small blood vessels again. And this is what we do with the robot. Again, a trauma to the foot that needs reconstruction. We use the robot to connect the vessels. And here you can see the artery pumping that we connected. And here you can see the tissue on top of the foot, vital, and the problem is solved. Last part of our studies are hand injuries. So in the hand, nerves, tendons are very superficial. So glass injuries or tried opening avocados gives nerve injuries. And we can repair these nerves. You can see it. It's on the left is the finger and on the right is the hand. And we repair these nerves with the robot. But these are 1 millimeter in diameter. So all small structures. Last one is finger replantations. So high-powered tools like circular saws, they sometimes take off a whole finger or more than one finger. And we can put them back on the body, but you need to reconnect all these small structures. And that's what we use the robot for. And our first case was a couple of months ago when a patient amputated his thumb and we were able to place it back on the hand. So what did we learn from those studies? We learned that it's feasible and safe to use this new robot for microsurgery and for even the smaller super microsurgery. There is a learning curve, so it takes longer in the beginning, but the improvement is quick. And of course, larger studies are needed to really compare the robot to the hand and see the outcome for patients. Where is the robot useful and where should we use it? So my last part of the talk, future perspectives. So I've been talking about the robot and precision of the human hand, but our human eye can be helped as well, because microscopes are really powerful and you can see very well, but the microscope has to be above the wound, and the wound is sometimes very far away. So in awkward positions, we have to sit for long hours, giving back problems and neck problems to microsurgeons. So a way to solve this is to use powerful 3D cameras instead of a microscope. And you can see an operation where Professor van der Hulst and me are operating on a lower leg reconstruction, and we are connecting these small vessels while we're sitting freely, ergonomically, as we want to sit. And we can combine this with the robot, as we did here. Secondly, is to use the contrast agents to see structures that you don't see with your eye, like this ICG seeing the lymphatics. And if you connect that to the robot and to AI, you can have a sort of Google Maps showing you where to go. Go left, go right. And this will help. And lastly, we are creating a new robot. MUSA 3 it's called, as it is the third generation. And this combines the exoscope, so the camera system, with robotic precision. And this is a small, if it goes, a small video. It's not working. That's a pity. I'll try to go back. I'm clicking on it, but it's not working.
Is it not working, Tom? Can we need to continue?
So I'll continue because I want to thank everybody who's involved. This project is a crazy project of already 17 years, and we would never have succeeded without the help of all the people around. I'll give back the word to you, Prorector.
Thank you very much for this very nice presentation. Now the opposition will be opened by Professor Bouvy, professor at Maastricht University in Innovative Surgery.
Thank you. Dear candidates, I would like to congratulate you and your supervisory team for working so hard on this excellent Dutch robot for at least 20% of your life to help patients with improving their outcome. I wondered if ChatGPT was already known with this microsurgical robot. So I asked him which are the most important microsurgical robots. And I was pleasantly surprised that I found your robot at the second place. The Da Vinci robot was on the first place, and then we had the Mako system of Stryker, the Artemis, the Zeiss Kinevo 900, but I do not think that it's a real one, and then the Axis. And then I asked which one of these is the most sustainable robot, taking care of the environment. And I would like to have your answer on that.
Okay. Highly esteemed opponent, thank you for the question and for taking the effort to ask ChatGPT. It's very modern. I didn't dare to use it while writing this thesis, although it was tempting. I think that our robot will be the most environmental friendly of those that you named. We had the opportunity that the Da Vinci robot came first. And we saw that it was not only too large for microsurgery, but a lot of doctors were also complaining about the disposables, because you can only use the tools a couple of times and then you have to throw them out. And this is a way that this company can make money, but this was very irritating the doctors who were operating with this machine. So that was one of the concepts that we had a total different route, and we thought we click in the reusable tools that we already have in our hospital. Secondly, I think our robot is very small and lightweight, so for energy use and draping of the robot, you don't need as much. And I think also for the OR personnel and the OR time that is needed, I think a small robot that we use is much easier and will be lower in our how do you say that? Carbon dioxide emission.
Yeah, thank you for making this robot so sustainable. And indeed, I think you're on top of this list. I would like to go with you to chapter 5. That's a randomized pilot trial with a design which is interesting because you, if I'm right, you compared one single microsurgeon operating either with the robot or manually.
Yes, correct.
And so if you go to page number 166, in your dankwoord, you say, in my eyes, this is probably the best microsurgeon in Europe. In this setup, do you think that it's wise to compare your robot with the best manual microsurgeon?
Well, thank you. That's a good question. Yes, I think from an ethical point of view, if you try something new, the patients who participate, they deserve the best care. So that was the reason that we didn't want to start with less experienced microsurgeons. So the first trials, the patients would be sure that if there was any problem with this robot, Dr. Kishau could solve it. Also learning the technique and seeing where the robot is beneficial or not in an experienced hand is something we wanted to learn. And because it was only a small pilot study, we wanted only one surgeon, because if you have 5 surgeons, you don't know what you're measuring. I think, on the other hand, this is not the best way to see the potential of the robot, because we know that robots, from our previous studies, in the early studies, you can see that people who never operated before, or people who have a very shaking hand and are really bad at microsurgery, if they use the robot, their skills improve very quickly. So I expect in the future that if less experienced surgeons will use the device, that the learning curve will look more promising. But as said, our ethical point of view in starting this new technology had priority.
Yeah, I understand your thoughts now.
Very brief.
Okay, last brief. So I think it's brilliant that in such an early stage of developing this robot, there was really connection between technicians and physicians in the hospital, because then you develop a medical need. In your future perspective part of the thesis, you elaborate on what will be next. And just one, there are 9 different things. Can you tell me the most important thing you want to develop in the next generation, MUSA 3?
Okay, thank you. Well, it's a real pity that the video doesn't work. I hope we can show it later, because what you can see, the difference between MUSA 2 and MUSA 3 is that, that the MUSA 2 is fixed to a ring. You can see this robotic arm and they hang on a ring.
Only a short answer. What is the next medical need?
Range of motion is the short answer.
Thank you.
Very disciplined. Thank you so much. The opposition will be continued by Professor Mani, professor in plastic surgery of the Uppsala University Hospital in Sweden. Welcome also here in the Netherlands.
Thank you very much. It's an honor to be here. Thank you, the promoters and co-promoters, and the family and supporting team that's here today. Mr. Candidate, I have a couple of questions. It's an impressive journey that you made together with the team here over many, many years. And of course, there's a number of steps along that journey that makes a difference. And I would like for you to reflect upon what's the most important one along this journey, which had the biggest impact on the outcome of where you are today.
Okay.
From a scientific point of view.
Okay. Thank you very much, highly esteemed Professor Mani. Thank you for coming over all the way from Sweden for this defense. Your question is, what is the most important part of the whole journey?
Yeah, or important steps that's made a difference here.
Sorry?
A step that's made a difference for you.
A step that made a difference. Yeah. Well, as said earlier, I think the biggest step is the incorporation of the technical world and the medical world. That is I think the most important step. There are so many devices that being invented that are not really useful to a lot of people, and people try to sell it to doctors, and that's the reason why doctors don't like the industry. That's the way I was trained. And I think from the start that we asked the technical people to solve our problem, and these people took the effort, like Raimundo Cao, to come to our operation rooms instead of discussing on Zoom or talk, but they really came to the operation room, and we really went into the cellars of the technical university to understand each other's world, and then start creating something. I think that's the basic, the fundamental, most important part.
And now that you've hopefully making a full researcher by the end of this day, you will most probably have the possibility to be a promoter, co-promoter yourself, and I'm sure there will be new ideas that needs to be addressed and innovations and such. How will you set that up, having had your learning journey behind you? How will you address that?
Well, yes, we've learned a lot from history and from the first studies. By writing this book, I had to read more articles than I should normally read. And I learned about the ideal concept of introducing robotics in medicine. And I think by coincidence or logical thinking, we did the right steps. So we started in preclinical, fake vessels, animal studies, then a small study. We had to randomize from the medical ethical committee, which I didn't understand, in 10 versus 10 patients, but I'm glad they forced us to do it because we can continue this. And I think the next step will be to have larger randomized studies, because I really believe in keeping the randomized studies instead of showing what a robot can do. Multiple trials, multiple centers, multiple operators.
That takes me on to the next question, which is now if we are to be implementing this specific robot, how would we go about doing this in a fast, appropriate and safe way?
Thank you. That's
and how do you get science into that?
Okay. Yeah, that's a very good question. We tried one center out of Maastricht, which was your center, so you know all about this. It's hard work to get everything right. I think the most important part is first to conclude what are the endpoints of our studies. So together with other centers, first we need to take into consideration we have to do exactly the same, otherwise it's useless. So from a scientific point of view, I think it's very important that on beforehand we agree on protocols and collecting the data and follow up.
The opposition will now be continued by Professor Selber from the USA, freshly arrived yesterday, also professor in plastic surgery of the Cornell Health Institute.
Sorry. And congratulations, esteemed colleague, on this work. I've been watching you go at this for about 11 years, and nice to see it manifest in this process for you, so congratulations for that. As those of us who have been working in robotics and microsurgery for a while, there's some discussion around the economics of the problem. the economics of the problem we're trying to solve, but also the economics around some of the solutions when they're more technically complicated, such as in robotics. Can you talk a little bit about the economics of lymphedema as a problem and the economics of robotics and this particular robotic platform in trying to kind of solve that problem?
Okay, highly esteemed colleague, professor, thank you for coming over, getting a jet lag for only 2 days of sitting here together with us, because I think the people who don't know you, you're one of the founders of the Robotic Microsurgery Society, and you've done a lot of work on pushing the limits of where other people stop in a good way. So in lymphedema, it's a relatively new field. So lymphedema treatment has been tried in the 1950s, 60s already, but they abandoned it because it didn't work. And in retrospect, that was because our techniques, so our microscopes were not powerful enough, the instruments were not small enough, and the techniques were not refined enough. So now lymphedema treatment is growing rapidly because we know how to do super microsurgery, which is the smaller type of surgery, below 0.8 millimeter vessels. And more people are learning about this technique now, so it's increasing. Lymphedema, if you don't do anything, is a lifelong problem. And well, in Holland, every 1 in 6 or 1 in 7 female is confronted with breast cancer. So it happens a lot. And although our treatments are getting less aggressive with axillary clearances, still a lot of women receive therapy to the axilla. And I think lymphedema is a very common problem that is not really recognized everywhere. And this is a lifelong problem, as I said, because the only thing you can do is you can have drainage therapy for physiotherapists or compression garments, which help a bit, but you get a stigma with a compression garment, and you can have recurrent infections. So if you add all these costs of a lifelong treatment and you compare it to the cost of a robot and an operation, as long as it works, it's worth the effort. Now, then robots will bring extra costs. So the question is, how good are we with our hands doing these operations? And I think some people are able to do it, but even the best don't always have their good day. And I think a robot will standardize the precision that we have on a good day or on a bad day. So patients know the quality is the quality. So I think robots will give more surgeons the opportunity to do this very precise work, but also the surgeons who are doing it, they will be better. And because the outcomes of lymphedema surgery is relatively new, so we don't know a large amount of patients yet, but it's building up. So there are randomized clinical trials everywhere now, and we're learning more and more. So who will benefit from the operation, who not? Sometimes it's too late to do it. Sometimes it's too early. But the costs should be comparable. And of course, studies should prove it. So we need cost analysis studies incorporated in the clinical data.
Thank you. And I hope you will continue and do that work as well. You mentioned training more doctors. You mentioned standardizing. Can you speak a little bit to, and I know that you're an educator in addition to being a clinician and a scientist. So talk to me a little bit about how we bring new physicians onto these types of platforms. Is there a thought about how to train them formally, even a curriculum design perhaps that we have considered in this process?
Thank you. That's a good question, and I know you're working hard on that. And we have thoughts about it, but we haven't incorporated it yet. So, of course, anyone who has to who will work with the robot has to be trained. And not only the surgeon, but the whole team around. So the OR personnel, everybody needs to know how does this machine work. And in the early days, that was a concern to me, because the first studies we do, if they fail, everybody says it's a shitty robot, it doesn't work. So we trained the OR personnel, we trained Dr. Kishau, Professor van der Hulst. They all did 3 days of learning to know the machine, learning how it works, but also practicing. So in the general plastic surgery program, robotics is not an issue yet. It's so small that it's not standard, but it is growing quickly. I think you're ahead of time, and we have to develop this. So in our unit, with generation 3 coming up, we are already planning the studies, and so we do everything in the studies, but we are already planning the training. So first, you use silicone vessels. Then rats are usually used, and the question is we try to lower it down, because I think on rats, you don't need to have rats, because if you consider the artificial intelligence coming up, I think you can do virtual reality training, which is much better than training on humans or training on animals.
So you've led me to my next and final question, which has to do with sort of the future of all this work. Mm-hmm. And we have the robot, which is now the body, and we are still the brain. But as these machines begin to learn and develop some of these competencies, how do you see that converging with the purely electromechanical work of operator effector systems like the robot?
Okay, yeah, that's a big question, and that's a question a lot of people have. So patients usually ask, is the robot operating by itself? That's the first question that you get. I think AI is scary, but AI is also really good, as I said, to simulate training. So you can have augmented reality training before you touch a patient and get experience. Second thing is, if we train other people, we are saying, oh, it's too slow, or that's too fast, or don't squeeze so hard. And it's a subjective analysis of if someone doing a good job or not, and if you're learning yourself too. So robots, the good thing of robots, it's mechanical, as you say, so every force and every movement is registered. And with the use of AI, you can collect all these data and really get feedback to the one who's doing the operation, saying, hey, you were doing this repetitive movement all the time. Why did you do it? And this gives very, very good learning. And for patients, it's also good because if we, like you say, if we connect the robot to the brain, which is big data, the robot can tell us much more than we see now. So if you measure all the things that the patient has, like the heart rate and the temperature and the blood pressure, and you connect it to the operation registry, you can already see, oh, this is not going well before anybody knows. And also postoperatively, which patients recover early, which patients get infections. It can all be related to each other. So I think AI will really learn us a lot in the coming years. And lastly, about operating by itself, I think a microanastomosis is a very predictable technique. It doesn't move a lot. So I think in the near future, it will be possible for AI with robots to do parts of the operation automatically.
Thank you.
Thank you. The opposition will now be continued by Professor Ruurda, professor in invasive robot-assisted surgery from the University of Utrecht Medical Center. Welcome.
Thank you. Dear candidate, first of all, I want to congratulate you and the team with the extensive work that you've done. I think it's really admirable that you brought it so far with this design and clinical trials and every step in between of this new robot. And it's even more admirable that you've been so productive in the meantime to produce the next generation of robotic surgeons that take the whole first row here. And I hope they can follow a bit in English, but I guess they do. And I was triggered by a couple of things you said already. First of all, you specifically said, and you also stated on page 12 of your thesis, that the Da Vinci is not designed for microvascular surgery, even though it was designed to reduce tremor and to scale motions. Can you allude a bit more on that and what the Da Vinci might need to become good in microvascular surgery?
Thank you, Professor Ruurda, highly esteemed opponent. Thank you for coming, and thank you for your question. The the question that you ask has been a frustration of your neighbor for about a decade, because the Da Vinci robot is actually a really good robot, I think. It's just too large and too powerful for microsurgery. So there are small instruments on the Da Vinci, like the black diamond forceps, and they are capable of holding small needles. So in theory, it's possible, but it's much more difficult. You have to be careful. And Professor Selbo already performed even super microsurgery with this Da Vinci, but it's really hard work. It's not easy. A second drawback of the Da Vinci, besides the power and the big tools, is the optical magnification. So if we go really into small vessels, we need really powerful microscopes. And it's about 30 times magnification. And the Da Vinci can go up to 10, if I believe. And with a lot of exoscope camera systems as well, like I showed, the magnification and the quality of the image is not good enough to see the real details that you need. So that was the second thing. So for a Da Vinci machine, it would be a better camera, much more power, and smaller instruments. And then I think it would be possible.
That seems like a direction that is easier to go than developing a whole new robot. And I think a scope suited for microscopic surgery is very, I think it's on the shelf, and instruments made smaller. And I thought you would say you needed force feedback.
No, we don't yet. That's an extra nice thing to have, which we don't have in microsurgery. So when the Da Vinci came, a lot of general surgeons say, hey, I don't feel what I'm doing anymore. In microsurgery, you don't feel what you're doing. You see what you do, but it's too small and fragile that you cannot feel. So the force feedback, if you have it, it would be perfect. And you can feel these small structures. But if you design it and it's not entirely right, it's disastrous because you don't know what you're doing anymore. Yeah. And secondly, I think I don't agree that the Da Vinci console is the way forward for everything, as it is. If you look at costs, as the question was there, it's a 500 kilogram machine. It's made for endoscopic surgery, so it does have advantages, because I didn't talk about it, but if you harvest these flaps, as we call it, so you take out the vessels, sometimes we need to make big incisions to get there. And with the endoscopic possibilities of the Da Vinci, harvesting of flaps is done by Professor Selbo already, reducing the donor side morbidity. But for the pure, for example, lymphatic surgery, which is really precise, small incisions, superficial, I don't think you need a big Da Vinci machine in your operating room.
No, and I agree with you. I think you will have certain specific robots for specific indications that will do better than a generic robot. Yeah, and saying that, you've designed this robot, you've tested it in a lab setting, then in a rat setting, and you did worse in time, you did worse in performance, subjective, but still worse. How did you approach the ethical committee to saying, I do worse, it's actually not so good, but still we want to do a clinical trial? I would not be very enthusiastic if I was in that ethical committee to have you move forward.
Yes, that's a good question. I think.
By the way, you have said that a couple of times now, but it's not up to you to judge our questions, but it's up to us to judge your answers.
Okay, thank you for that. The committee, I think if you play safe, that's the most important thing, being a doctor. But if you only play safe, you won't progress. So we have to take decisions where the logic is there, that you can move forward. But you have to do it in an ethical way. You have to do it in an open way. And that's why we showed that it was worse. Because everybody knows if you do something new compared to something what you're used to do for 10 years, it's slower.
So if you say this, how did you inform your patients? We're going to try something that is proven to be worse on you now.
No, I didn't have to say that, because our outcomes and clinical outcomes were comparable. So it's slower. So for me, the concern was that the time that it takes to use a robot...
You didn't know the clinical outcomes when you informed the patients, because this was the first in men.
Yes, but we were there, and I was there at every operation, so I could see if things go wrong, I wouldn't proceed, of course. So for myself, I knew. Your question is, how can we convince the medical ethical committee? And I think to show, because we saw that the times were good, were longer, but our early outcomes were promising. So they were equal, and they're still equal. And maybe in larger groups we can say it's worse, but so far we cannot say. So because it takes longer, I expected much more problems in the beginning of infections, wound infections, being wound open for longer times usually gives more infections. So it's really happy that even though it took longer, the outcome was the same. So I think if you look at the learning curves, which are only 10 attempts, we didn't use a lot of patient or a lot of trials to get better.
So in hindsight, because if I look at your outcomes, they're equal, but are you happy with these outcomes? Do you think this is good enough?
Yeah, I'm never happy, and it's never good enough, and that's the problem that I have. When I was in this company, I always wanted the robot to be better. Yeah. But technicians said, okay, we have to stop now and test the robot. So it's never good enough.
So there's a bit of external pressure to go into clinical practice. That's what I hear in between the lines.
No, it's not. It's the only way to learn is to use it. But you have to use it in an open way. And sometimes things are not better, but then you have to show it. Yeah. Because you will learn and you will improve. So the bottom line is, it's pure logic. Precision and microsurgery and robotics are such a logical combination that it's just a way of, it's not a question if it comes, the question is how it comes.
I don't disagree with that, and actually I follow you completely, and I think this is the way forward. But I think you might have considered developing the robot a bit further, doing more lab tests in order to overcome your learning curve, because there is a clear learning curve, and I think it's not finished yet, before you go on and move to a human experiment. Mm-hmm. And that's what I would have said if I was in that ethical board. I would say, practice for another year, go on, develop the robot a bit further, and then try it on real life subjects.
Yeah. That's a good comment. Sorry, I couldn't judge it anymore.
But make it fun.
But I think you will always have this learning curve again, because it's something new. So no matter how good this robot is, it's new.
And that actually brings us to the question that was posed before. How do you train people so good that you don't put your patients at risk?
Yeah, that's a good question, and that's what I sorry, I'll stop. But that's absolutely true. So the clear answer is, I think as a surgeon, you know if you're good enough, and you know if something's working or not, and that's the bottom line. I mean, you have to show to everybody, and you have to write down protocols. But the real ethical question is in your own brain, in my opinion, and you have to prove it and you have to show it. But there is no straight line. So for us, we used an average score that before. So I designed my own question. So before we went into human studies, we had asked Dr. Kishar to perform this microsurgery, and we stated ourselves this has to be proficient level. So it was judged by other blinded microsurgeons, and if we didn't reach a level, we wouldn't proceed. So that was the line we draw ourselves. But it's just.
So you state you're good enough to do this,
yes.
And the ethical committee agreed on that.
Yes.
And do you have any clue, if I still have 1 minute, on how you will even get better?
Well, first of all, Gen 3 of the robot is coming, because the second generation is fixed, and all comparable robots and all competitions are fixed that we use in microsurgery. And this robot will have the opportunity to have unlimited motion. So I think the technique itself will improve our performance, and structured training will help. And as lastly, if you design these studies with other doctors in other centers, you collaborate, you discuss, and you learn from each other. And I think that's an important way of improving your learning curve quicker.
Thank you very much for the nice interaction, and
Thank you very much. Thank you very much. We're not there yet. We have a final opponent, Professor Wim van Gemert, pediatric surgeon, professor at our own university. And we know that he is able to continue for at least an hour. So the floor is yours.
Microphone.
Also my first time that I have 10 whole minutes to find out questions. So also my congratulations with the wonderful work and also for the supervising team. First of all, I heard you saying you're never happy. You should be, because you say your glass is always half full, which means you're an optimist. And you need that when you are exploring new innovations and want to implement them. Okay, so first I want to start with a benign teaser, which is, I think, because of the sound and a bit of playful competition between different specialisms in medicine. So you're stating several times in your work that microsurgical anastomosis requires great technical skills. Well, in chapter 3, you use two surgeons, one experienced microsurgeon, one resident in training, one medical student. If you look at the graph, the medical student has the same outcome in time and probably also quality. So, is it really so difficult? Do you really need this technical skill? Or did you have a brilliant, you were unlucky that you have a brilliant student with great motor skills?
A highly esteemed opponent, thank you for the question, and also thank you for answering it for me. Well, it's difficult to say, of course. There are only 3 people that have been judged, different levels, and it's random if you have a good or a bad person in this scheme. Yeah, I think the younger generation will learn quicker in robotics, but the younger generation also doesn't want to stay in the hospital the whole life, all day, in the weekends. And I think microsurgery is a difficult technique to learn because you're not only learning your skill, you need to master your tremors, you need to master your mindset, but it's not only the trick of doing the anastomosis, it's the whole setup of preparing the vessels. And that's a very challenging operation, because if you squeeze one time too hard in these vessels, you're out. And I think it isn't mentioned yet now, but another advantage of robots and younger generation learning it more quickly. So you don't need the experience of a microsurgeon to learn the robotics. That's what we learned from a learning studies. And I think that's a solution for the problem that we face, or a problem that the younger generation doesn't want to make the hours that I think is sometimes needed to get this expertise level.
Okay, thank you. Then I want to go on with chapter 5 again, about the setup of this randomized trial. And I was wondering why you chose this method, because you are comparing the LVA, the lymph venous anastomosis, handmade or with a robot, and you randomize them. And your primary outcome is a difficult one. It's lymph ICF score, which we know is very close together. If you would do a power analysis, which you didn't do, I think, you would need enormous amounts of patients, so it will not be possible actually. So with this setup, you know on beforehand that you will not have significant differences, in my opinion. Secondly, your randomization process, I don't understand exactly. It's a block randomization, but you have 5 blocks.
4.
Oh, I read 5. That's why you have, from each 4 patients, that's why you have 12 in one and 8 in the other. So it's a little bit uneven, okay. And not completely comparable. Why not do a non-inferiority study, for example? You need less patients, and you can at least show that you will be at least as good as the gold standard, the hands-on anastomosis, or just a prospective study.
Okay, yeah, thank you for the brilliant question. I wasn't allowed to say it, so I say it one more time. Well, a couple of questions. So the randomization of the people who got robot operations or hand operations was 4 block randomization. And it should have been 10 versus 10 patients. But in 2 patients, the robot didn't work. So one patient, the robot was not working, and in the other patient, the toolbox that was needed fell on the floor and was unsterile. So we had to change from robot to manual surgery. So that made the differences in 12 versus 8 instead of 10 versus 10. So that's not because of the randomization. And we included them in the hand, although it's not the intention to treat principal, but these groups were so small that we should have... we couldn't. And then the other question about the study itself. So first of all, in my mind, I had the same idea of just showing that it's feasible to do these operations and just follow them and register everything. And when the Medical Ethical Committee asked us to make it into a randomized trial, I also didn't understand, because I thought these groups are so small, this will never be significant, whatever we do. So it's really pseudoscience. But what we learned from it, and instead of doing other studies, is that we collect data now in a very structured way, and we hope we can do power analysis, and we can look at which outcomes are better usable for the next studies. So I hope we can do that
You need more centers and trained surgeons, yeah, sure. Just a general question also for me. What I always think with this operation, after women with breast cancer had operation, had radiotherapy, it will not only be the lymph vessels, 1 or 2 lymph vessels which are damaged, but it will also be more tissue in that area and the surrounding tissues. So I think even if you make a perfect anastomosis, of course it will help, but it will never reach 100% improvement. Am I correct or not?
Yes, I think you are correct. I think if we damage something, you damage something, and scar tissue is scar tissue. So it's never the situation before. What we learned, though, is that we try to do preventive surgery now. So instead of just doing axillary surgery and then look the other way and see what happens, we're starting to do prophylactic reconstruction of these lymph vessels in the axilla. That's a literature growing now. And also, as you say, if you reconstruct the breast, it's not only the shape of the breast, we are also trying to connect nerves when we do breast reconstruction to give the sensation back to the breast. So, of course, there is a lot of damage and a lot of things need to be repaired, and we also can improve every day.
Okay, thank you very much. One question. It's not a difficult question, but it's about somewhere you sum up the areas which the microsurgery can be used. Urology, cardiac surgery, all kind of stuff, but not pediatric surgery. Which is strange because we are not far away from you, but everything is mini, maybe not micro, mini. And we discussed about it before, thinking about, okay, what could be areas within pediatric surgery where microsurgery could be used. So maybe you thought of this more than I did, so please elaborate.
Thank you. Well, you are seeing more patients in pediatric surgery and doing more operations, so probably I'll miss some. But the indications we already thought of is to make arterial venous shunts for children who need dialysis. You create a connection of the artery to the vein, and you can puncture it so a dialysis patient, kidney patient, can be helped. And a lot of times this gives problems because they clot. And that's the first, indication that we find. So
Yeah. Thank you very much.
Good timing. Thank you very much, dear candidates. The committee will now withdraw and will assess the quality both of your thesis and your answers. So you can now further reflect upon the brilliant questions that were posed by the committee without us being present. You can even maybe show the video if it works. And we will be back after our deliberation.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Hit the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. South side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
maar hier staan. Hier staan, ja. Kun je meegaan, ja?
Dear candidate, Tom van Mulken, the degree committee here present has assessed the quality of your thesis and your defense. And in view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the title of doctor. Doctor. How nice is that? And Professor van der Hulst is authorized to confer upon you this academic distinction in accordance with Dutch university custom. And I invite your supervisor to now take the floor.
And he will do that in Dutch. Is that good for you? Oké. Belooft u dan dat u altijd volgens de beginselen van wetenschappelijke integriteit te werk zult gaan? Dat is eerlijk, zorgvuldig, transparant, onafhankelijk en verantwoordelijk?
Ja, dat beloof ik.
Krachtens de bevoegdheid dan ons door de wet toegekend, volgens het besluit van de commissie hier tegenwoordig, verklaar ik hier bij u Tom Johannes Marie van Mulken tot dokter te bevorderen en u alle rechten te verlenen die daaraan volgens wet en gewoonte zijn verbonden. En ten bewijze hiervan overhandig ik u nu de bul door rector, secretaris en de overige leden van de promotiecommissie ondertekend en met het grootzegel van de universiteit bevestigd. Mag ik? Dokter van Mulken. Oh, Miemelaar. 17 jaar heb je daarover gedaan. Piemelman, dat moet ik van Marleen zeggen trouwens. Nou, het is niet helemaal eerlijk. Het is 12 jaar, want je bent pas echt begonnen toen je uit Australië bent teruggekomen. Dus het valt eigenlijk nog best mee, 12 jaar. Maar het begon eigenlijk nog iets eerder, 20 jaar geleden, al in Maastricht. Toen zaten we bij een sollicitatiegesprek ergens op het Onze Lieve Vrouwenplein, als precies te zeggen in Hotel d'Erlon. Want de staf had na een hele week werken op vrijdag geen zin om de sollicitatiegesprekken voor een ANJOS nog ergens in het ziekenhuis te doen. Dus we dachten, en volgens mij was dat ook Marleen en ik, van nou, laten we dat dan maar in Hotel d'Erlon doen. Ik weet niet of we dat tegenwoordig nog zouden kunnen doen. Maar goed, dat ging goed en jij na een biertje werd je aangenomen. En er is heel veel veranderd dus in die tijd dat jij inmiddels gepromoveerd bent. Maar je ging vrij snel in opleiding. Je was een tijdje ANJOS, daarna ging je in opleiding en je was al vrij snel gefascineerd door het robotverhaal. In 2006 was ik begonnen met die Da Vinci. En dat lukte niet echt. Ja, het lukte wel, maar ik vond het niet echt een succes. Maar we kregen wel door veel media-aandacht toen. En dus ook kwamen we in contact met de TU Eindhoven en met Maarten Steinboes en Raymond de Cauw. Werd er toen het idee geboren om een specifieke robot te maken voor microsurgery. Je hebt er al eigenlijk iets over verteld. Raymond ging aan de slag en het eerste prototype werd ontwikkeld. Thomas bemoeide zich er aanvankelijk nog een beetje mee, ikzelf. En jij ging even naar Australië en kwam terug en je beet je er helemaal in vast. En uiteindelijk werd de Musa jouw muze. En inmiddels, moet ik zeggen, heeft Elon Musk zijn eerste menselijke robot al gemaakt. En is er ook een Italiaanse robot ontwikkeld. Maar wij waren toch stiekem de eerste. En wat dat heeft laten zien is dat die ontwikkeling van robots in combinatie wat nog gaat komen, AI, ook niet meer te stoppen is. En anderzijds heeft het jou ook jouw doorzettingsvermogen laten zien. Want ja, al die tijd ben je er toch mee bezig geweest. Maar dat is wel een beetje gek. Want eigenlijk ben je helemaal niet zo van de gadgets en zo. Je bent eigenlijk veel meer van de retro. En ik dacht, wat is nou die connectie En eigenlijk kwam ik erachter, wat je hebt gemaakt eigenlijk is een moderne pantograaf. Ik weet niet of je dat kent. Ik heb hem voor je meegenomen, dus die wil ik je ook maar meteen even geven. Dat is eigenlijk, ken je dat nog van vroeger? Daar past nog veel meer bij eigenlijk. Hè? Daar ben je waarschijnlijk ook ooit door geïnspireerd. Dus ik zou zeggen, ga daar nog even mee aan de slag. Hoe dan ook, je deed dat onderzoek naast je klinische baan als plastisch chirurg met aandachtsgebied handchirurgie. En samen heb je ons geholpen om het hele team op te houden. Dus we zijn echt een A-team, een DA-team zijn we eigenlijk. Met regiomaatschap Figurie, Laurentius, Zuiderland, Mooikliniek en het MUMC, waar je met name dit onderzoek hebt uitgevoerd. Maar je bent natuurlijk ook een enorme mimelaar. En dat was ook nodig voor die ontwikkeling van die musa, want daar heb je geduld voor nodig. En dat is iets wat Jan en ik iets minder hebben, denk ik. En dat mimelen heeft ons goed geholpen, want anders waren we niet zo ver gekomen. Maar het mag af en toe ook wel eens iets minder. Hè? Met name als we al 3 maanden hebben gewerkt aan businessplannen en analyses hebben gemaakt, zulke stukken hebben geschreven en gaan vergaderen. Van gaan we het nu doen of niet? En jij zegt, zullen we het toch nog een beetje gaan uitwijken? Misschien moeten we dat toch iets minder doen. Ja, en over mimelen gesproken, je dankwoord is ook bijna net zo lang als je hele proefschrift. Dus dat zien we wel terug. Maar goed, het is goed om in ons A-team een mimelaar te hebben die een beetje kritisch blijft. En dat doe jij. En het is ook goed om af en toe iemand te hebben die een beetje weerstand geeft. En je hebt ook zo'n baldadigheidsknop, hè? Die heb je vandaag nog niet aangezet. Ik verwacht dat dat dadelijk nog gaat komen, maar ik ben blij dat je het net niet gaat. Maar goed, ik dwaal af. Hè? Terug naar die micro. Dat zelfs die kleine dingen zoals microchirurgie impact kunnen hebben, dat bleek wel, want we werden opeens uitgenodigd bij De Wereld Draait Door. Nou weet ik niet zeker of dat aan de robot lag, of dat ze in Hilversum hadden gehoord dat wij een A-team in Maastricht hadden en dat ze graag de face in de uitzending wilden. Dus dat kan natuurlijk ook nog. Maar in ieder geval, wij zaten daar en we waren toen al bij de DWDD grensoverschrijdend aan alle letteren. En inmiddels bestaat de hele DWDD niet meer, omdat je zo lang over je promotie hebt gedaan. Maar goed, je reist nu wel de hele wereld over en miebel je een hoop bij elkaar. En je bent zelfs in het bestuur van de Internationale Vereniging voor Robotmicrochirurgie. Maar naast dat klinisch werk, dat bestuur en onderzoekswerk, heb je natuurlijk ook nog een gezinsleven. En of een echtgenoot en 3 kinderen nog niet genoeg was, moest er natuurlijk ook nog een coronahond bij, Bobby. En het lastige van Tom is, hij is niet echt een multitasker, hij is een typische monotasker. En dat schijnt op zich ook beter te zijn voor je brein, maar goed. Dus als de hond naar de oppas moet, dan moet die robot en ook de operatie maar even wachten. En dan komt dus de hond eigenlijk voor de robot. En dus zou een robot thuis misschien toch ook wel handig zijn. Dus ik zou je toch nog eens even aanraden om naar dat filmpje te kijken van de Optimus 3. Hè. Die heeft Tesla vorige week geïntroduceerd. Die kan de hond ook uitlaten. Dan kun je gewoon 's ochtends naar het werk komen. Tom, je toewijding aan je vak en familie is echt wel inspirerend. Want je hebt natuurlijk voor die passie voor je werk ook passie voor thuis en liefde en zorg voor je gezin. En dat is echt een indrukwekkende combinatie waar je trots op mag zijn. Vandaag valt er een last van je af, hè? Waardoor je misschien wat meer speelruimte krijgt en eventueel nog een paard erbij kan kopen of zo. Of misschien kun je toch eindelijk weer eens een keer gaan sporten, want ik zie langzamerhand wel dat er wat vet bij is gekomen natuurlijk. Nou, ik kom nu bijna aan het eind van mijn verhalen, dus niet bang zijn. Maar dat doe ik niet voordat ik je nogmaals feliciteer. En dan wil ik je ook je familie, Maartje, Max, Bente en Sophie, en je vader feliciteren. Je moeder was er ook graag bij geweest, maar zoals je zelf zegt, ze is er in jouw gedachten zeker bij. Dus ook familie, vrienden en collega's wil ik je allemaal hier bedanken, ook dat ze aanwezig zijn. I also would like to thank Maria Mani and Jess Selber for coming all the way from the USA and Sweden and to attend this meeting. Sorry I had to do this laudation in Dutch, but I will translate it for you with ChatGPT. And dus nogmaals wil ik je hartelijk feliciteren. En ik wil u allen vragen om een applaus te geven voor de grootste mimelaar en onze nieuwe dokter in de robotmicrochirurgie.
Nou, dokter Miemelaar zou ik bijna zeggen. Dokter Van Mulken, van harte gefeliciteerd. Ook namens het college van bestuur en namens de raad van bestuur. Ook hier vertegenwoordigd met Gabriel Zwart van het MUMC. We hebben normaal gesproken een geheimhouding bij wat er in de raadkamer gebeurt. Hè? Dus dat kwartiertje dat wij ons even terugtrekken. Maar ik kreeg net ook ingefluisterd, je mag er wel iets over zeggen. Dus nogmaals, niet alles, maar wel een aantal woorden die zijn gepasseerd. Hè, aangezien jij ook de vragen van de commissie ging beoordelen, gaan wij dan nu ook jouw antwoorden publiekelijk delen. Een paar woorden die werden gezegd, flawless. Dat is een hele mooie beschrijving van jouw antwoorden. Brilliant, at ease. En als het over het klinische werk ging, dan zag je ook volledig jouw passie en jouw enorme diepe kennis. Dus veel complimenten vanuit de commissie voor de manier waarop je zowel het proefschrift hebt opgeleverd, maar ook de manier waarop jij de verdediging hebt gedaan. Dus je bent er vanaf. Goed gedaan, van harte gefeliciteerd, nogmaals. Beste mensen, wij zijn daarmee aan het einde van deze academische ceremonie gekomen. En ik ga dat dadelijk symboliseren door keihard met die hamer te slaan. Maar voordat we dat doen, een paar huishoudelijke mededelingen. Wij zullen dadelijk als cortège samen met de net benoemde dokter en zijn paranimfen en zijn familie als eerste de zaal verlaten. Dan gaan wij een foto maken. En dan willen wij u vragen om vast naar de refter te gaan, om een glas te pakken. En dan zullen wij zo snel mogelijk ons bij u voegen. Veel dank voor uw komst en tot zo direct aan de borrel. Dank.